Hello, I am Lee Spector, and this presentation is Evolution Evolves with Auto-Construction for the 6th workshop on evolutionary computation for the automated design of algorithms at the Genetic and Evolutionary Computation Conference in 2016. This is joint work with Nick McPhee, Tom Helmuth, Maggie Casal, and Julia Knox. What I'm going to talk about today is first tell you what auto-constructive evolution is. Then I'll briefly survey some prior work on auto-constructive evolution. I'll focus then on recent developments in auto-constructive evolution and show you the results of some of this recent work. I'll end with a quick discussion of the prospects for work in this area in the future. So what is auto-constructive evolution? It is one idea for how to evolve an evolutionary process while evolving solutions in an evolutionary computation system. So the general context here is that we're using evolutionary computation to solve problems, but while doing so, the evolutionary mechanisms that drive the evolutionary process themselves evolve. There might be several ways to do this, and the way that it is done in auto-constructive evolution is that individuals in the evolving population are responsible for producing and varying their own children. So the methods by which they produce children are encoded in the individuals, and those methods are subject to variation and selection. Getting this right requires understanding something about the evolution of variation, and it turns out, I believe, that that's quite an interesting subject that may help us to not only build auto-constructive evolution systems that are very useful, but may help us to understand evolutionary computation and evolution more generally. But the hope is, indeed, that it may produce evolutionary computation systems that are more powerful than we can currently write by hand. Now, this is a 15-year-old project, auto-constructive evolution, and it builds on older and broader-based ideas within evolutionary theory and evolutionary computation. And yet, at the present, I guess I would assess the utility of auto-constructive evolution as being something like genetic programming, but harder to do and less successful in what it's produced so far. Now, that may make you question why we're still focusing on it after 15 years. And there are a couple of reasons. One is that we believe that ultimately, the evolution of the evolutionary mechanisms is going to produce better evolutionary computation systems than we can devise by hand. But also, some of the recent results, especially embodied in a system called AutoDog, which I'll be discussing today, are for the first time being able to solve some significant problems and are demonstrating intriguing patterns of the evolution of evolution. So I want to show you those results and show you the patterns that we're beginning to see. So, in general, evolutionary computing looks like this. You start with random instances of the kinds of things that might be a solution to your problem. You assess them all. If it's a very easy problem or you're very lucky, you find a solution already, but more likely you haven't found a solution. So you select parents, you produce children that are varied from the parents, and then you reassess all of those children to see if you have now found a solution, and if not, you go around the loop again. Genetic programming is simply this same model where the random instances you begin with are programs. They're assessed by running them, and when you succeed, you end up with solutions that are programs, that are software. Now, in genetic programming, as ordinarily performed, variation is accomplished by means of mutation and crossover operators, and indeed sometimes we allow programs to produce a child that is identical to the parent for the next generation. We sometimes call that straight reproduction. So this is what it looks like in traditional genetic programming, and the important thing to note is that the mutation and crossover steps are written and configured by humans. By contrast, in auto-construction, it is the program itself which is executed to produce the child, and the way in which it produces the child is encoded in the program itself. And there may be access to other programs for mating, for recombination, but it is this first parent that you might call the mother that produces the child, and the code of that program is what is itself doing whatever kind of mutation, crossover, variation will be done. And this is a little bit more complicated when genomes are distinguished from programs, as they will be in some of the work I show you a little later today, but this is the basic picture. So, in autoconstructive evolution, individual programs make their own children. In doing so, they control their own mutation and recombination rates and methods, and in some cases, mate selection and so forth. Because of this, the machinery of reproduction and diversification, which is the machinery of evolution, itself evolves. In PUSH, which is a programming language I'll tell you about in a moment, experimentation with autoconstructive evolution is easy and natural. Okay? And before I get into PUSH and show you how we actually do it, I need to mention there are a couple of obvious hazards when conducting autoconstructive evolution. One is cloning. If you get guys who are pretty good, so they get selected as parents a lot, and they clone themselves, then you can quickly overrun the population with clones and have no potential for future evolution because there is no externally imposed variation. There are these other kinds of programs that don't quite clone themselves to make children, but I call them dead-enders because the children do not vary in significant ways. And so while they're not identical, they're behaviorally identical with respect to problem-solving performance and offspring production. And this is another way to kill evolution, is to have a population full of dead-enders. Error catastrophes are also always a hazard, and that is just when variation in these evolved entities is too extreme, and there's not enough heritability so that the descendants no longer solve the aspects of the problem that their ancestors. Now, how do you actually do this? Well, the PUSH programming language was developed for programs that evolve, and it is used mostly for regular genetic programming with hand-designed variation operators. However, it was really motivated in its first development to allow programs to simultaneously express solutions to external problems and to express how their offspring should be constructed in the same integrated program. And the key idea is that it's a stack-based programming language in which data flows via stacks, not via syntax. And what distinguishes it from other stack-based programming languages is that there is a separate stack for each data type, such as integers and floating point numbers and so forth, and that there are stacks for code. And the execution stack of the program itself, as it is executing, it is available for manipulation. And it is this code manipulation and execution stack manipulation that makes PUSH particularly powerful. It has a very simple program syntax, summarized in this first line. It is a Turing-complete language with rich data and control structures. And if you develop a genetic programming system in which the programs that you're generating randomly and then varying to evolve solution programs are PUSH programs, then we'll call that system PushGP. And there are quite a few versions of PushGP that have been developed over the years. It makes it easy to combine the manipulation of programs or genomes with other computations because of the way in which different data types are handled on different stacks. Now, in fact, in modern PUSH, as of the last couple of years, we also have genome representation, which is separate from program representation I showed you in the previous slide. It's a linear representation in which instructions are listed in the sequence, and the structure of where sub-expressions start and end are encoded both in metadata on the instructions themselves and in epigenetic markers, we call it close markers, that say where blocks of code end. We also, with this same representation, have epigenetic silencing markers, which allow for different kinds of mutations. That's an interesting topic for another day. What this does is it facilitates the useful placement of code blocks, allows for other techniques like epigenetic hill climbing, which Bill ECADA has worked on. But mostly for the sake of autoconstructive evolution, it makes it particularly easy for a program to manipulate programs, genomes for children, as linear structures, so that we think that facilitates this autoconstructive evolution process. All right, so to conclude about PUSH, it's out there, it's available, there are implementations in many source languages. The work described in this presentation all uses Clojush, which is a PUSH implementation in Clojure. I want to very briefly run through some prior work in auto-constructive evolution. The first system is from 2001 called PushPop. And in this system, while you're evaluating a program for how well it solves a problem, it also leaves what you might think of as eggs on a code stack. And then later on, when there are tournaments among parents, there are these tournaments for hatching rights, and the auto-constructed eggs are born at that time, unless they're cloned. This was a very open-ended system in which the programs could access and run code of the entire population, both while they're solving the problem and while building babies, which they're doing at the same time. Yeah, 4 years later, a system called Swarm Evolve 2 involved the evolution of behavior, including reproductive behavior, in a 3D virtual world controlled by evolved programs. Auto-construction was followed by imposed but program-controlled sort of cosmic ray mutation. There was all sorts of facilities for color sensing, energy conservation, communication, energy sharing, and so forth. Then in 2010, we came back to problem solving and looked pretty hard at when can it solve problems, how can we make that better, how can we analyze how it works. To that end, we made it asexual and separated reproduction and problem solving in different execution phases of programs. And we imposed constraints in selection and birth. And I want to mention some of those constraints here, since they'll relate to some of the current work as well. We preferred parents with non-stagnant lineages, looking for a change of performance in the most recent half of the lineage. And we prevented birth from lineages with constant differences from each generation between them. Some of the work in Autodog is not quite these same constraints, but these ideas fed into our current work. In 2012, Kyle Harrington primarily and some other collaborators developed new auto-constructive evolution systems using zipper data structure for constructing children and a different set of constraints on reproduction, requiring, for example, neutral or better errors of children, and apply this to synthetic problems that involve program structure. Now, looking at all this prior work, it did demonstrate that selection can promote diversity, and it exhibited interesting dynamics of diversification and adaptation, but it all had weak problem-solving power, and it was difficult to analyze the results or compare it to other genetic programming work. So, here we are in the present, Autodog, 2016. It's different in many ways from the prior work, but these are the four highlights of how it's different. We construct genomes rather than programs. We have distinct modes, phases for construction of offspring versus problem solving. We select parents combinatorially, not on aggregate error. I'll say more about all of these in a moment, but that's an important one. And we enforce what we now call diversification constraints, and we're still experimenting with that last element. So what is constructed? In prior work, executing a program would construct a child program, but in Autodog, executing a program constructs a plush genome, okay, which we saw previously. To facilitate this, we need a genome stack, and we need genome manipulating instructions. That's very easy to add in push. This happens to be a set of genome manipulation instructions that we used in the runs described in the paper. How and when is it constructed? Well, in prior work, sometimes it happened during error testing, sometimes problem inputs were available when doing it, and so forth. In Autodog, we just have a different genetic operator, like crossover and mutation, but we only use that one genetic operator, and when we use that genetic operator, it runs the parent program and produces the child. So that is separate, that is during the reproduction phase, not during the problem-solving phase. And when we run it for reproduction, for construction, the inputs are no-ops, and when we run it for error testing, the random instructions produce constants rather than non-deterministic values. Now, who gets to construct? How are the parents selected? Well, in prior work, we used what everybody uses for most GP, which is error aggregating methods like tournament selection. In Autodog, however, we use lexicase selection, and this is a relatively recently developed parent selection algorithm in which the errors on multiple test cases are not aggregated into a single value that is then used for tournaments. We don't aggregate the errors on multiple test cases at all. Instead, what we do every time we want to select a parent is we shuffle all the test cases. For the first test case, we keep all of the individuals who are best at that test case. then we repeat with the next test case, and so on. And we do that until only one individual remains, and we use a different shuffle for every parent selection event. What results is that the parent selected might be a specialist in whatever cases occurred near the beginning of the list for that random shuffle, and may or may not be particularly good on average, even though it may contribute to the evolution of generalists later. Okay? Now, I don't have time to talk about the results of Lex case selection in general. However, I would suggest that you go to Tom Helmuth's presentation in the GP Best paper track later in the conference, where he talks quite a bit about Lex case selection. And the bottom line is, at least on many kinds of problems, and this is the front page of a paper we have on IEEE transactions and evolutionary computation, the bottom line is that for many kinds of problems, we see significantly and large-scale improvement in problem-solving power by using Lex case selection instead of an aggregating selection method. And we also see higher levels of behavioral diversity. All right, so last distinctive feature of Autodog Who Survives. Well, in prior work, we had a variety of constraints on survival, including prohibiting only clones, and also including some of the kind of constraints on progress within lineages that I mentioned earlier. Well, in Autodog, we only allow those to survive that satisfy diversification constraints on reproductive behavior, which we determine from a cascade of temporary descendants. So the idea is we only want guys to survive who will diversify their descendants. And that is the constraint we seek to apply to a cascade of temporary descendants. And you could have a variety of different diversification constraints. What we use here is that children must differ from the parent, and they must differ differently from each other in the ways that they differ from the parent. And we apply this to the programs expressed by the genomes of the children, and we enforce it in the cascade with only 2 children, so it looks like this. We're trying to decide whether this one labeled parent here, I know that's a little confusing, but that guy, we want to see if that guy is going to be allowed to survive into the next generation. It must have been produced by some parent previously. And to see if it can survive, we execute it twice to produce children, and the parent-child differences must be positive and must not be the same as each other. So again, this idea of the diversification constraint is still under development, and the big issue we're trying to decide is how can you tell if an individual has the potential to produce diverse adaptive descendants, and we have considered larger cascades, and we've considered variation also of genomes, and also the reproductive behavior, and also problem-solving behavior. And it's useful in thinking about what we're trying to do here, to think about the requirements for the evolution of evolution. And some different terms here, the way I use them anyway, is that diversity is that individuals vary. Diversification is that individuals produce descendants that vary in various ways. And that's what we're using here. But there's a sort of more ambitious idea that we're playing with various versions of, which is recursive variance, that individuals produce descendants that vary in the ways that they vary their offspring. So the results that I want to show you come from software synthesis benchmark problems. We've proposed these at Gecko 2015, and PushGP in its normal configuration can solve most of these. Here I'm going to focus on the replace space with new line problem. These are all problems that come from introductory computer science textbooks. And in this one, you're given an input string and you want to print the string replacing the spaces with new lines, and you also want to return the integer count of the non-white space characters. So that's 2 things that the program has to do, and it also has to deal with multiple data types. Here is a simplified solution in Push. And PushGP, under some configurations with normal genetic operators, can succeed up to 95% of the time. Autodog doesn't do nearly as well, but it does sometimes solve the problem. And that in itself is significant, considering that we are evolving the evolutionary system along with evolving solutions. Give you a sense of the kind of results we get. These are the same figures that are in the paper. This one shows the parent-child differences when ordinary genetic operators are used over a successful run on this problem. And what you see that I think is not surprising is that after a burst of sort of wild variation initially on, we get into a mode where the amount of difference between parent and child is relatively stable and is relatively evenly distributed from no mutation, no variation, up to some maximal level. So we see this dark band of all of the parent-child differences over the run. They all occur sort of below this Levenshtein distance of 200, and it's pretty stable across evolutionary time. By contrast, here's what the same thing looks like over one of our auto-constructive runs. And what you see here is a lot of, well, you don't see an even distribution at all. So you see reproductive methods producing certain parent-child differences that remain stable but are coexisting within a population that produces children in very different ways. And you see this unfold over time to produce different regimes of parent-child construction happening at different phases of the run before a solution is finally found. The ancestors of some of those guys at the end may have been produced with some of the wilder, larger distance methods earlier on. And there's a lot of interesting complexity going on here. The main thing I want you to show is it changes over time, it is richly structured. I would say that it evolves, and I would say that since the variation is the essence of how evolutionary computation works, the fact that the way variation changes, the way that variation happens changes, the way that parents and children differ changes, shows that evolution evolves in auto-constructive evolution. Similar kind of graph, this one is not for parent-child differences, this is for program sizes over a run of non-auto-constructive evolution on this problem. And here is the distributions of problem sizes across a run with auto-constructive evolution. And you will note that there are some strong similarities between this and the graph that we saw earlier. They're fairly related. And there's a lot of interesting structure here as well. This is really an anecdotal piece of evidence, but you can see that there's some reasonably dramatic differences between these graphs of the ancestors of solutions in successful runs, single successful run with standard operators, and a single successful run with auto-construction. So the conclusions and the prospects. Auto-constructive evolution can now solve reasonably hard problems, at least some of the time. So far it takes longer than standard genetic programming because it must evolve evolution along with solutions. Can it solve problems that can't be solved by ordinary genetic programming? Possibly, because it evolves. It has not yet, though. But studying how and why it works may help us to improve it, and studying how and why it works may also help us to understand the evolution of biological evolution. I would like to thank the members of the Hampshire College Computational Intelligence Lab for very stimulating discussions, anonymous reviewers who are quite helpful, Josiah Erickson for system support, and Hampshire College and the National Science Foundation for their generous support as well.